can load that gel, you need to mix your sample with a sample loading buffer. And so what is inside these buffers? Well, there's something colored so that we can see the progress of the run, make sure everything's going okay, know when to turn off the power. It's got something heavy so our sample doesn't just pop up out of the well. And if we're running a denaturing gel, it's going to have something to unfold the molecules. So if you're running a protein gel, a denaturing protein gel, like an SDS page, the SDS is this detergent. It's going to unfold the proteins. If you're running a nucleic acid gel, you're probably going to have formamide to denature the nucleic acid, so pull apart the DNA strands from one another and unfold any RNA hairpins and things like this. And there might be other things. You might have a reducing agent for a protein gel. You might have EDTA in your nucleic acid gels buffers. So let's talk about what's inside all of these. And yeah. Gel electrophoresis is awesome. It allows us to separate molecules such as proteins and DNA and RNA by their size. And it does so by using electricity to get them to swim through a gel mesh. And this mesh can be made up of agarose, it can be made up of polyacrylamide. And the meshier it is, the slower the molecules are going to go. But not all the molecules are going to travel at the same speed. The longer, the bigger the molecules, they're going to get tangled up more in that mesh. And so they're going to travel more slowly. And the reason why they're traveling through the mesh at all is that we have an electricity gradient set up so that there's a positive charge on one end, and the molecules, we make them so they're negatively charged or they're naturally negatively charged in the case of the nucleic acids, and then they're going to be motivated to move towards that positive charge. But we don't want them to move all the way out of the gel. Instead, we stop the gel running when they're midway through, and then we look using some sort of stain to see where the different molecules are. And this allows us to get a sense of the composition of our mixture of molecules, as well as do further things like a Western blot if you want to identify a protein that's there, or some sort of Northern blot or Southern blot for look for nucleic acids, all sorts of different things. But in order to do this, we need to get our samples into that gel, and we need to be able to know when to turn the power off. Both of these things are going to be aided by the use of a sample loading buffer. So this is just going to be that thing that you mix with your sample before you stick it into the wells. And so the wells of the gels are going to be, this is a polyacrylamide gel, and so you can see it kind of has these tall and skinny wells. For the case of an agarose gel, like this, you're going to have kind of like these deeper wells that you're going to stick your sample into. And so there are different formats of gels, and we'll talk some about how different buffer components are compatible for one versus the other. The biggest difference we're going to see when it comes to these buffers is going to be whether we're doing a denaturing, and so we're actually unfolding the molecules, or whether we're doing a non-denaturing or a native gel where we're kind of not unfolding them, we're just letting them travel as is. So say if you had DNA, double-stranded DNA that would stay together, if you had in a non-denaturing gel, but it would come apart in a denaturing gel. Protein complexes will stay together in a native gel, but come apart in a denaturing gel, and they'll also get unfolded. And so because the shape of the molecules can impact how they travel. And so when we do a SDS page, say that's typically what we do for proteins, that's going to be a denaturing, where the denaturing agent, the unfolder, is going to be this molecule called SDS. And so we'll get much more into all of these details, but this is where we're going. So our buffer is going to contain a dye. And so sometimes we just call these like a loading dye instead of a loading buffer. But they might have multiple dyes. These are going to allow you to track the gel's progress, know when to turn off that electricity. The dye also helps you see your samples while you're loading them, which is a huge help, especially when you're trying to locate those wells. And it can help you see if there are problems that happen during the run. And so, for example, the other day I was running this gel and I started to see that the bands weren't running evenly and there was something weird happening. And it turned out that there was some like bubble on the electrode or something. And so I was able to, by adding a little more buffer and by kind of tapping things out, was able to mostly get things fixed. Another thing that you can see is that your gel might start smiling. This is often caused by it kind of like heating up too much if you're trying to run it too fast. If you see it start smiling, then you can basically turn down the voltage, but you can only see that that's happening because of that dye front. And so the dye is really helpful for that as well. They're going to have something heavy in there to help your sample sink and not just pop out of the well. Then they might have a denaturant or not. And so remember, the denaturant is going to be what's going to unfold the molecule, what's going to remove the structures, remove any interactions between molecules. And so sometimes you want that, sometimes you don't want that. And depending on what you want, you're going to include or not include a denaturant. And what type of denaturant you include is going to depend on the type of molecule. So for a protein, you're typically including SDS. And for nucleic acids, you're typically including formamide. For protein, you also might add a reducing agent to break up disulfide crosslinks. So this would be something like DTT or BME, beta-mercaptoethanol. And so let's look a little more into the details. So starting with that dye or dyes, there can be multiple dyes in the sample loading buffer. For a protein gel, for like an SDS page, you typically just have a single dye, and the single dye is usually bromophenol blue. Now, it's really important that you remember that this dye is not showing you where your proteins are. All it's showing you is where the dye front is. So this dye is going to be smaller than the proteins, at least most proteins. It's going to travel smaller than them. It doesn't travel exactly the same as proteins. So it's not like, some proteins actually might travel faster than the dye, but that doesn't mean that they're actually smaller than the dye. It's just that it also depends on the charge and things like this. I should mention that an important thing about all of the dyes that we use is that they're going to be negatively charged because they need that negative charge to travel through the gel towards that positive end. And if they had a positive charge, they would travel backwards up out of the gel, which would not be good. And so basically, though, we'll see this in the case of nucleic acids too, the dyes are going to run different in different types of gels. But the dye front, typically what we do is we choose a gel percentage. So you can basically adjust the meshiness of the gel by choosing a higher or lower gel percentage. So with a higher percentage, you're going to be better at separating small things. And with a lower percentage, you're better at separating big things. And you choose a gel so that when the dye front runs off, you're going to have your molecules are going to be in the center of the gel. And that's where you're going to get like the best resolution. And so typically that's how you choose the percentage. And then once you've chosen that percentage, you can know safely that your sample is going to still be in the gel when that dye front runs off. And so that tracking dye is going to help you know when to stop it, but it's not going to actually show you where your sample is. I should mention that sometimes you run like a ladder that's pre-stained. And so in that case, you will see bands for different proteins, but that's only in that ladder. That's only in that marker lane with those pre-labeled proteins. Your proteins are not going to be labeled. And so your proteins are just going to be somewhere, hopefully above the dye front. And so then when the dye front runs off, you can then stop the electricity running, stop the molecules from being motivated towards that other end. And although they're not motivated towards that other end anymore, they can still kind of diffuse, so they can move around randomly. That's going to have a hard time diffusing out of the gel because the gel is kind of like holding them into place. But if you give them long enough, they'll wiggle their way out. So you want to make sure that you're doing some staining right away or whatever you want to do. And then, so you stain the gel and now you can see your bands. And so you might stain it with something like a Comasi blue. And then remember that the dye in the gel is typically a Comasi blue. I mean, it's typically a Bromophenol blue, but it's just the tracking dye. Speaking of Bromophenol blue, this is often one of the dyes that's included in a nucleic acid loading buffer as well. But with nucleic acid sample buffers, you often have multiple dyes in there. And what's cool about these is that they kind of allow you to get a sense of the different size molecules that are in the gel. And so if we think about the nucleic acids, we can have these multiple dyes. And each of these dyes, because they have different sizes, they have different charges, they're going to travel differently. And they're going to travel differently in different types of gels and different percentages. And we can, if we know what size they migrate at, so what size of nucleic acids do they travel along with? So for example, if you're running a denaturing polyacrylamide gel, so this is one of those denaturing, so we're going to be separating the DNA strands from one another. We're going to be erasing the secondary structure of RNA, things like this. If we had a piece that was about 50 nucleotides long and we ran an 8% gel, we would expect it to show up in between the bromophenol blue bands and the xylene cyanol bands. If we were to run it in a non-denaturing polyacrylamide gel, and we ran that same 8% gel, it would now be potentially overlapping with the bromophenol blue dye, and this could be blocking your signal. In this case, you might want to actually leave out the bromophenol blue and make a loading buffer that only had xylene cyanol, because this is going to be a lot higher. It's not going to interfere with the signal that you're seeing. You're not going to get your bands kind of being shadowed out by the dye. But this wouldn't be very helpful potentially because it's going to run like it's a much bigger. And so by the time that you turn off the gel, your sample might have run out. And so often there's actually a third dye that's included, such as amaranth, this like pink one. And so you just need to go and look up the different running percentages for different gels. Like what's that, the dye migration charts. And so you can find those online, as well as buffer recipes, and we'll get into some of that. But so here's an example of a DNA loading dye, this Easy Vision. I really like Easy Vision. This is not a paid endorsement or anything, but what's really cool about it is that unlike most sample loading dyes, it actually has a DNA binding stain, a fluorescent stain in there, that's actually going to bind to the DNA in your sample, and then you don't have to do like a post stain with CyberSafe or ethidium bromide or anything like that. You don't have to add that to your gel. It's already in the loading buffer. But most loading buffers don't have this. So you do need to add that DNA stain, that fluorescent stain to whatever, to your gel or to the buffer afterwards, like soak it. But if you're using this Easy Vision, it's in there. But you're not going to see that when it runs. Instead, you're just going to see these different dyes. And so there's actually different versions of Easy Vision. So I'm using Easy Vision 3, which has the 3 bands. And this is what you can see here. You see that xylene cyanol, you see that bromophenol blue, and then you see that amaranth. They have different versions with only like 2 dyes, so without the amaranth or with just the amaranth. And so, yeah. So and again, it's going to run differently on different samples, but that's kind of what it would look like. Okay, so that was the dye, but there's more in the buffer. And one of the most important things in there is they're going to just be something heavy so that your samples sink into the wells when you're loading them and they don't just pop out, because that would be really, really sad. And in fact, if you have like ethanol in your samples, maybe you were doing some sort of nucleic acid precipitation and it had ethanol and you didn't remove that ethanol well enough, well, now your sample is going to kind of float out of the wells, and that is not good. So you need something heavy, because even if you don't have ethanol, your samples would still be like the same density as the water. And so it would kind of just diffuse out, and then it would get into all the other lanes and it wouldn't be traveling through your gel straight, and it would be really, really sad. So what you want to do is include something heavy so that your sample sinks. This something heavy is often some sort of like sugary molecule. A common one is glycerol. A common thing is glycerol, so this like polyol thing. So you want to include something heavy. Commonly, this could be like glycerol. It might be Ficoll, like Ficoll 400, which is basically this big sugar molecule. It's a bunch of sucroses stuck together. And so it's got like these linkers and it's kind of sticking together all these different sucrose units. Basically just something to make them heavy. If you're using like a formamide dye, which we'll look at in a minute, for which denatures nucleic acids, that's going to be all thick and heavy on its own. And so you don't need to add something heavy in there. Okay, but yeah, so you want to make sure your sample sinks into the well and it stays there while you're loading your other lanes, because you're not going to have them motivated to move through the gel until you turn on the electricity. But remember, diffusion is able to get the molecules to move around, but the heavier they are, the harder it's, and the thicker and stuff, the harder it's going to be for them to move around. So that viscosity is playing a role there, helping us. Okay, so now we need to get to that decision point. Do we want to denature our samples? And so remember, if we denature things, we're going to actually be separating the molecules from one another. So if you had like protein complexes, they would be separated. They would be separated from one another. And if you had the individual proteins are all going to be unfolded. And so sometimes what you want to do is you want to run a native page where you're basically keeping the molecules together. You might be doing this to identify protein complexes and things like this. But most of the time when we're doing a gel for proteins, we're doing what we call SDS page, where SDS is going to be the detergent that's going to allow us to denature our samples. And so I should mention too that denaturing basically is going to unfold and take apart the molecules, like from one another and unfold them. But it typically does so with the help of heat. So we typically boil samples when we're denaturing them. This is going to kind of help them unfold initially, and then the denaturant is going to stabilize them in that unfolded state. So sodium dodecyl sulfate is going to SDS, is this negatively charged detergent molecule, and it's kind of going to coat the protein once it's unfolded. It's got this long lipidy tail, so this like greasy, oily tail, and this hydrophilic or water-loving head. And what's going to happen is that the protein, normally when it folds up, it does so that its hydrophobic parts, its parts that water doesn't like to hang out with, are hidden in the inside of the protein. If you unfold that protein, well, now those parts are going to be exposed to the water, and water doesn't like them. But if you have an SDS molecule that's going to be globbing onto those parts and hiding them from the water, well, now the protein can stay unfolded and stay soluble because it's got that hydrophilic part, that negatively charged head of the SDS. This negatively charge of the SDS is also really important for SDS page because it's providing proteins with a negative charge. So proteins may or may not have a negative charge already, but there's going to be inconsistent between different proteins. And we want them all to have a negative charge and an evenly distributed negative charge so that they're going to be traveling on the basis, they're going to be traveling in the first place towards that positive charge. And two, they're going to be traveling based on how long they are. And so SDS is going to do both these things for us. When it comes to nucleic acids, we don't have to worry about the charge because nucleic acids, so like DNA and RNA, they have this negatively charged backbone already, and it's evenly distributed throughout the molecule, which makes it really great. So we don't need to add the negative charge, but we do need to add something if we want to unfold the molecules, remove secondary structure, as well as make double-stranded like DNA or RNA, make it single-stranded. And so we often think about nucleic acids as kind of just like these wiggly strands, but really, especially RNA, it can fold up into these really complex structures. We call this like secondary structures, like things like hairpins. And if we want, those are going to impact how the molecules run through the gel. So we can erase those shapes basically in different ways. And one of them is we use formamide in the loading dye, which is basically going to break up the hydrogen bonding. So it's going to break up the interactions between the different bases that are holding the strands together and holding the different shapes together. And it's going to kind of take the place of those and satisfy the needs of those nucleic acids without having them bind to other nucleic acids. And so we typically include formamide in our loading dye. And this is also going to make it heavy, so we don't need to add glycerol or something like that. Often we then use, if we're doing a denaturing polyacrylamide gel, we also include another denaturant, urea, inside of the gel. And this is going to allow it to then stay unfolded throughout its trip through the gel. Okay, so that was the denaturing, but denaturing is not all you might need to do if you want to totally get a protein unfolded. And so you might also want to add a reducing agent to your buffer. Now, a reducing agent, what it does is it's going to break up disulfide cross-links. So it's going to break up those cysteine-cysteine bonds between different molecules or different parts of the same molecule. These are covalent bonds, so they're going to be stronger than just the attractions holding together different molecules and holding together the shape of the molecules in most cases. These disulfide bonds, though, they're not as strong as the bonds holding together the different amino acids into that chain. And they have a vulnerability that we can use a reducing agent, something like DTT, dithiothiazole,
or BME, beta-mercaptoethanol, or TSEP to break up those bonds. And so often if we want to both denature and reduce our proteins, and so we can do this by including a denaturing agent, something like BME, into our sample loading buffer. We often add this fresh, or we make the rest of the dye, and we make a lot of it, and then we take aliquots of it. So we take portions, and then when we want to use it, we add BME, and then at that point, we store them in the freezer, whereas we can store the rest of it just at room temperature. And we need to do this because that reducing agent is kind of going to like oxidize and go bad over time. And so we can use these different dyes. We can use different denaturing or non-denaturing conditions for both nucleic acids as well as for proteins. So with proteins, with denaturing, we're talking about an SDS page typically. And if it's non-denaturing, we often call it a native page. For nucleic acids, we're often doing a non-denaturing gel, such as just like a normal agarose gel or a TBE gel. But we might do a denaturing gel, such as a urea page. And in that case, we would have formamide in our loading buffer. There could also be some other things inside of a sample buffer. Sometimes they contain like a pH buffer. So commonly, like your SDS page loading buffer, it might have tris in it, which is this pH stabilizer. You might also have something like EDTA in a nucleic acid gel buffer. EDTA is basically a chelator, so it binds or it like bites down on a metal, such as like magnesium, something like this, that is needed, and those metals are needed by nucleases. So these some nucleases, RNase A is resistant, which is annoying because that's one of the main really ones that we worry about chewing up our nucleic acids. And it's not dependent on metals, so we have to be really careful. But other nucleases can also cause issues, and they're going to have to use a metal. And so if EDTA is kind of biting around that metal, then it will hide it, and then the nucleases can't use it. The EDTA can also kind of hide excess metals that might be affecting how the nucleic acids are going to be running through the gel. EDTA is also the E in the TBE and the TAE, in case you were wondering. Okay, so those are the main components of our loading buffer. We have the tracking dye or dyes. They're going to help us know when to turn the gel off. And they're also going to help us know that we're actually loading our sample into the well. So it allows us to actually see that sample that we're loading rather than loading a clear liquid into clear liquids. So really helpful. We might include something. It also includes something heavy, so your sample is going to sink down into the well, not just pop right back up. It might contain a denaturant so that the molecules are going to unfold. They're going to, complexes will come apart, this sort of thing. You might also have a reducing agent in there to break up disulfide crosslinks in the case of proteins. And then you might have some other bells and whistles like a pH buffer or EDTA. Most of the time, these buffers are going to be given in terms of relative concentrations. So it's going to be like something like a 6x or a 4x or a 2x, which is basically, in the case of a 6x, it's 6 times more concentrated than you want to use it at. If it's 4x, it's 4 times more concentrated, 2x, 2 times, etc. And so, for example, if you have a 6x loading buffer, you would have a working concentration would be 1 microliter per 6 microliters total. So you would add 1 microliter per 5 microliter sample. If you had 4x, you would have 1 microliter per 4 microliters total. So you would have 1 microliter of the dye or the buffer and 1 microliter of your sample. If you had 2x, you would have 1 microliter per 2 microliters total. So 1 microliter of your sample and 1 microliter of the buffer. But of course, you can also make more than you need. And more on calculations with relative concentrations in another post. As for actual recipes for the dye, I'll also provide some of those. So this is like an SDS page loading buffer. So you can see it's got that tris. It's got that SDS. So remember, the tris is the pH buffer. The SDS is going to be our denaturant and negative charge provider. The glycerol is going to make it heavy, and the bromophenol blue is going to make it blue. And then you can add the BME fresh. For nucleic acids, I was making these the other day, and they were just so pretty, which kind of motivated this post. And so basically, you can have denaturing or non-denaturing. If you have denaturing, basically, this is going to include something like bromophenol blue. but it's not going to include something like formamide. If you have formamide, it's going to be heavy, it's going to be all viscous, and you can actually see it here. And so yeah, it's viscous, so I don't have to add like sugar or anything. But this is the non-denaturing, and it actually has this Ficoll 400, so that sugary thing to make it all viscous, and so you still get really pretty. Yes, this is the xylene cyanol one, also, and this is the bromophenol blue. So you can see that this one's kind of darker and this one's kind of tealer.
I recommend that you dissolve everything you have to dissolve other than the dye before you add the dye. And because the dye is going to make it hard to see if things have dissolved. So if you need to dissolve that Ficoll, that like sugary stuff, you're going to want to dissolve that first and then add your dye. And so then afterwards, you also want to syringe filter this probably, and then you can make aliquots and store them.
So I hope that helped you, and have fun loading your gels and making sample loading buffers if you're doing that, because it's really pretty.